Welcome, everyone. We will start in around 4 minutes. So, thank you so much for coming. Today, it's about our guest speaker's work, how he approaches rejuvenation and tries to solve aging. And he also formed a company around his research to come up with treatments. And I shared the link in the chat. Um, and if you think this is interesting, please share the room with people that you think could learn from it. And we will start shortly. Thank you so much, everyone. Yeah, just to let you know, I shared some resources in the chat. First is the Cell Life information about our guest speaker's company, how he tries to address aging and how to make our aging easier or slow it down. And then also I shared the link for his paper in the chat, and on top is the presentation, so feel free to look into those resources while we are waiting for our guest speaker and for starting. Usually we start on top of the hour, and yeah, feel free to share the room if you think this is interesting for people that you know. And yeah, we'll start shortly. I think there's a lot of information in the meantime. So yeah, thanks for being here. If you have questions, if you want to participate in the discussion, feel free to raise your hand in the meantime and come up on the stage. So it will be easier to take your questions when you have them. Thank you. Yeah, in the meantime, while we are waiting for a guest speaker to come here, he is new to Clubhouse. Apologies for the little bit of waiting, but in the meantime, this is a really this is really exciting research. Just to refer to the paper here really quick while we are waiting. So I don't know if everyone here in the audience knows how telomeres are always associated with aging and the length of telomeres in particular. And once the telomeres are shorter, it's really difficult to go back in time basically and lengthen them, especially during aging. So, what our guest speaker here successfully did was an intracellular transfer of telomeres between cells, and it rescued T cells from senescence and promoted long term immunological memory. So, this is really a really big deal in the aging research field, that he was able to transfer basically healthy telomeres and improved rescued cells from dying and improved even their function, because what T cells do is they have this long-term immunological memory to fight off diseases. And not just that he could um, transfer the telomeres, they basically implemented themselves in the cell in a way that the cells could perform their healthy function again. So that's why this is a really big deal, and that's why I invited our guest speaker here. And with his research, as I said before, he started his own company, uh, Send Cell Life, and how he would like to achieve human rejuvenation is he wants to use a similar approach to green technology. Oh, there. Are
Hi,
you there? Hi,
hi,
hi. Perfect.
hi.
How
Sorry,
are you?
this is new to me. This is new to me. I'm very well. How are you? Thank
Good,
you for the
good.
invitation.
Oh, thank you for taking the time in your busy schedule. I was just talking a little bit about why your paper is so important, your research, and I started talking a little bit about your company to, inform people that just joined because it's a public room and maybe don't know anything yet about your research, to just give, like, pass some time. So, thank
Yeah,
you so much for coming.
thank you. First question for me is that can you hear me correctly? Because I mean we're having some issues with the connection. So is my voice clear?
Yes, I can hear you very well.
Okay, thank you. So
Perfect.
thanks
And
again for the invitation. My name is Lanna, and I have a laboratory at Centicell. We study here the longevity and the biology of human aging. And we are focusing on T lymphocytes that are important to control infections and cancers, and they have to undergo this task for decades. So that's why they're a very good model to study biology of longevity, right? Because these cells have to persist in a memory state for a very long time. There is the issue of senescence that arises after progressive stimulation, at least is thought that arises after progressive stimulation of the lymphocytes, and immune senescence appears, and that leads to the deterioration of the immune response, leading to the aging process. And now T cells are started to be regarded as really at the pinnacle of all the aging phenomena across the body. And so that's very exciting for us at Centicell because we are focusing on delivering new medications, first-in-class treatments to rejuvenate the T cell state. There's been a lot of work on many different states of altered T cell function, such as, for instance, energy and exhaustion, but very little is known of the pathways that govern senescence in the immune system. And people sometimes they think they're the same thing, like senescent and exhausted T cells. Actually, they couldn't be more wrong. They're very different states, and they need a separate therapeutic intervention. And that's what we are trying to do. My company at Centicell. It's important because we target senescence that, obviously, as I said, appears as we grow older. But this is not just a phenomena of the age, but actually happens in a lot of different situations, inflammatory conditions, and cancers, HIV infections, and so on. So this is the slide on what is immune senescence, essentially. So moving forward, the hallmarks of immune senescence, there is loss of costimulatory receptors, there is lack of telomerase activity, there is reduced proliferative capacity, impaired metabolism, and so on. And over the past decade, work I've been doing has really characterized all these phenomena that go wrong in the T cells as we get older. They seem to be connected through a single pathway, which was previously unknown, which is called the SMAC. SMAC or SMACs are cestrin McKinase activation complexes. So cestrins are a family of poorly understood growth regulators and stress sensing proteins that accumulate during aging. There are three of them, cestrin 1, 2, and 3. They are found from worms to humans, so it's a very conserved pathway. In Drosophila, there is one cestrin, but then during evolution, they duplicated and they arose into these three different gene products. Actually, the name cestrins derived from little country, a town here in Italy. During a congress, the story is that people, when looking at their genes, they found homology in that. It was a country, a town, I think it was called Sestre. And so, because they realized that in that meeting, then they call these three different genes the cestrins. So the cestrins in the SMAC that I discovered act through a stress kinase or metabolic kinase regulator that is very well known, AMPK responsive protein kinase. And the AMPK exert its activities through three different effector kinases, ERK, P38, and JNK. It doesn't do it directly though, it doesn't show it here, but it does it through scaffolds. For instance, we know that P38 undergoes autophosphorylation upon activation of MPK and then binding to TAB1, and probably similar scaffolding proteins support the autophosphorylation reactions of ERKs and JNK proteins. What is very important and very cool about the SMAC is that essentially there are different roles of the different components in the pathway, meaning that upon a stress signal, the cestrins would appear, that they bind to the AMPK, and the AMPK induces the scaffold-supported autophosphorylation reactions, whereby each single MAP kinase here regulates a different aspect of the senescence of T cells. So, for instance, ERK would induce DNA damage in the T cell, P38 would arrest the telomerase activity, and JNK would suppress CD3 and CD28 costimulatory receptor signaling, and also intracellularly, it's the kinase, is a single kinase responsible for new characteristics of senescence I discovered during my PhD studies at UCL. I'm now professor at UCL as well. That is the loss of the TCR signalosome components of LCK, ZAP70, and all those proteins. So moving forward with the slides, then how the telomeres came into pictures. Well, obviously, as I said, telomerase is thought to be the single enzyme that is responsible for the aging of cells, including T cells. The story is that when telomeres get very short, there is a threshold around 4 kilobases at which proliferative activity stops, and then the cells enter a status which is actually metabolically impaired but still active. So it's not a quiescent cell, but is metabolically active because it produces compounds. For instance, it produces inflammatory cytokines and so on, responsible for age-related inflammation. And the senescence, therefore, has always been thought as a telomerase-related problem. So people, including myself, during my PhD studies, were really focused trying to reactivate the telomerase. However, there is an issue. There is an issue is that even if you overexpress telomerase or you overactivate the enzyme, and for instance, you do a simple experiment in which T cells are provided extra support with an extra telomerase activity. At some point, they reach proliferative exhaustion, and actually, they do that very quickly in vitro. And this is something and a paradox and a mystery that nobody in the field understood. Why a T cell that in the body can stay fit for decades, if that T cell is cultured in vitro, why that T cells last for only a few weeks and then essentially stop dividing? So it was clear that there was something missing to the story. So that led, so the next slide, to our new discovery now on Nature Cell Biology this month of the telomere transfer phenomena. So what is telomere transfer? So moving slide again, telomere transfer is an alternative pathway of telomere elongation that does not require telomerase activation in the T cell. When does it happen? It happened before telomerase is even activated. So telomerase is generally activated in T cells after antigen presentation, but you need several hours, a couple of days, to actually detect enzymatic activity in the cell. Telomere transfer often happens before. So when T cell in the lymph nodes during antigen presentation encounter an antigen presenting cell, those cells will undergo a decision whether they will become a senescent progenitor or whether they'll become a long-lived stem-like or central memory cell. So how does that happen? So when T cells, when an antigen-specific reaction takes place, in certain conditions, which we haven't yet revealed, APCs establish that that T cell is worth of a telomere donation. In that circumstance, what they do is they quickly degrade sheltering. Sheltering is a complex of proteins that protects and caps the telomeres at the ends of chromosomes. And upon sheltering degradation, there is an enzyme called TZAP, which was described to bind to telomeres that are devoid of sheltering proteins, and that would provide essentially the signal to cut and to eliminate, to remove, sorry, the telomeres from the chromosome ends. Upon essentially upon this activity of TZAP, then the telomere is packaged in a vesicle, and that vesicle is loaded with a protein called RAD51, which is an homologous recombination enzyme provided by the APC. That vesicle containing telomeres of RAD51 is transferred to the antigen-specific T cell that receive the vesicle and somehow incorporates these vesicles at some telomeres, probably ultra-short telomeres. We're now characterizing the pathways that actually is responsible for this fusion reaction and how the T cell decides where to incorporate the telomeres. It seems that the incorporation is not a stochastic process. Anyhow, the result of the fusion is that that cell is endowed with increased replicative capacity. It generates memory, so it's an antigen-specific, long-lived memory cell. It's protected from senescence because it's turned towards a stem-like state, and so on. So this is a very different thing than what we previously thought. We previously thought that senescence was essentially a consequence of repeated episodes of stimulation. And now, instead, we are proposing the rather different scenario in which before a naive T cell even start division, that cell is committed to either an aging fate or a stem-like or long-lived memory state, depending on a single event and a single decision whether that cell receives or not telomeres in form of donation from APCs. So moving forward with the data, the other slide, please. So the discovery of telomere transfer, a lot of studies, probably you've read the paper. Here I'm summarized some what I think is probably most significant things. One thing that during the reviewing of the paper we faced a lot of skepticism, and it took a long time, many years, to get the paper out, was that regardless of the methodology to study the telomeres, that is obviously standard fish, which is accepted obviously in the telomere field, but as an important limitation that obviously it's performed on fixed cells. So if you have to do obviously standard fish on fixed cells, you cannot study the movement of particles. And another aspect that we have used that actually produced similar results, we actually introduced the PNA TLC specific probes using glass beads that essentially they generate the transient holes in the plasma lemma of the APCs before encountering with T cells. So that is important because that allows us to introduce the fluorescent probes in the cell before forming an antigen-specific synapse. So in that way, all the telomeres, for instance, here in the 3D live telomere transfer experiment, I'm showing all those white signals, all those telomeres are obviously only of APC origin. So as I said, something that took a very long time to convince people and reviewers was that we were always observing these unusual, to use the reviewer's wording, unusual signals where these large clusters of telomere signals would appear at the synapse, and reviewers were thinking this would basically be the probe attached to the beads, which is a bit weird because the beads are actually visible at the naked eye, so that was impossible. Anyhow, we have demonstrated that obviously this is not due to an artifact induced by the beads, and instead it can be reproduced, as you see here in the top left, on a standard fish experiment. You can clearly see in the APCs, the CTB negative cell, the telomere in the APCs form these beautiful clusters at the synapse with T cells that precedes the transfer to the CTV blue positive T cell. Obviously, in the immune synapse field, it is very well known the polarization and clustering of molecules at the synapse precedes transfer in the opposite cell. We have performed experiments on synthetic bilayers that reproduce an artificial APCs. It's a big complicated experiments in which T cells are allowed first to form a synapse on these artificial surfaces such that they would release the T cell receptor in microvesicles on the bilayers, T cells removed, APCs offered those TCR coated bilayers, and then in that way introducing APCs with pre-labeled telomeres and vesicle proteins like CD63 here on the right, or PTH67, which is a lipid marker, we demonstrated that was actually the signal. The TCR released by the APCs was the signal at the synapse responsible to induce essentially the signal, basically giving your telomeres for the APCs to the T cells. So essentially, purified TCR microvesicles on coded B layers can trigger, as I show here, telomere transfer from APCs. So moving forward, obviously we had to demonstrate whether these telomeres would go where they should be. Essentially, would they go? Would they be at the T cell chromosome ends? And as you see here in the other slide, they do. So we proved this essentially um, probing DNA in the APCs with ADU, so then with reaction chemistry. After transferring these ADU labeled APC telomere vesicles to separate T cells that were not previously in contact with APCs, and again performing standard metaphase FISH, we demonstrated that those telomeres donated by APCs were found at T cell chromosome ends. This is very consistent both in humans and mice. It's about 8%, up to 10%, 8% of the recipient T cell chromosomes contain these telomeres of APC origin. The telomeres are transferred, as I said, in vesicles. These are just electron microscopy data that show the evidence of these. You can see this by transmission electron microscopy with telsicle, the labeling showing that most of the telomeres are inside the vesicle. Also, something interesting is that it seems that the amount of telomeres, as you can see on the gold labeling, seems to correlate with the size of the vesicle. And then also evidence on assorted, FAB sorted fluorescence activity vesicle sorting material by scanning electron microscopy for size determination. The size is that of an exosome-like vesicle in general. There are some vesicles which are larger, up to 300 nanometers, but that is a very minor population of vesicles. As I said, TZAP again is required for the telomere vesicle encapsulation reaction, meaning that if you silence TZAP in APCs and you stimulate APCs with a ionomycin, which is basically a signal that mimics the synaptic event of calcium release in the APCs for the synapse with T cells, release telomere vesicles, but if you block TZAP, the vesicles, so the telomere vesicles are not released anymore. Moving slides again, the transfer of TZAP can be observed at the immune synapse. If you transduce APCs with the fluorescent TZAP, they form synapses with T cells, and you can observe on a standard fish again. It's very cool co-localization of blue TZAP with telomeres at the synapse, demonstrating the transfer. Again, we went ahead showing that obviously TZAP is known to process telomeres that are that contain a low concentration of sheltering. So we reasoned that this reaction would occur at telomeres that have degraded or lost somehow sheltering. We demonstrated that upon activation by ionomycin in moving slides on panel A, or upon synapse formation with T cells in panel B. What you can see is that the sheltering levels here, sheltering is exemplified by two proteins in panel A, TRF2 and POT1, in panel B only TRF2, but the concept is the same. As you can see, 24 hours in the synapse, for instance in panel B, the level of TRF2, the green signal in the APC, which is the cells CD3 negative obviously, essentially is diminished, and this is a proteasomal degradation because when we pretreated the APCs with the proteasome inhibitor MG132, this essentially degradation of sheltering was prevented. Okay. Why this is important? This is important because this is the event that, moving slides again, that initiates the transfer of telomeres. So you can overexpress sheltering in APCs, and you can stimulate the APCs with uromycins, for instance, and you can precipitate the telomeric material from the supernatants. You can see quite cool the dot plot here, in which upon stimulation with ionomycin, in sheltered and overexpressing APCs, essentially there is no release of telomeric material within the vesicle fraction recovered upon ultracentrifugation. So the function of this phenomena is to generate, as I said, stemness and to protect T cells from senescence. And this is, moving slides again, what this set of experiments demonstrates. The T cells turn into a stem-like memory state within a naive T cell compartment of CD45 array positive, CD28 double positive T cells. You can see the increase 7.24 upon 15 days in culture with purified telomere vesicles of these cells, and within this naive double positive naive population, you can see the increase of stem-like T cells, which is a very important increase because we are talking about a very rare population of T cells. In general, these cells are about 1-2%, so we are able to boost it up to 10%. So that's quite significant. With a single treatment of vesicles in vitro, we are also able to remove senescent T cells and to remove cestrin and SMAC signaling and to expand them in culture. Uh, and you can see also the dysfunction on something that I will comment on later on, but these protective longevity effects are related to the RAD51 and are dependent to the RAD51 that is co-transferred in the telomere vesicles because in experiments when we have removed RAD51 in the APCs with the thironase, the right vesicles and treated T cells with those RAD51 depleted vesicles, the function and the boosting effect in terms, for instance, of proliferation or protection of senescence in the beta-galactosidase, as I say, is lost. So RAD51 is very important for that reaction. So does telomere transfer, moving slide again, happens in vivo? The answer is yes. We have designed experiments in which we've used the OT2 system, in which essentially an APC is preluded with ovalbumin injected into the foot pad of an animal. This APC has preloaded live labeled CY3 telomeres with the glass bead technology I mentioned before. 18 hours later, OT2 cells are injected in the caudal vein, and vaccination with OVAR is offered or not. And after 18 hours again, popliteal lymph nodes are extracted, and then essentially, obviously, the T cells that we have injected are labeled with CTB. And then when we recover the T cell material from lymph nodes of these animals, we can look at the CD3 positive cells that we have injected, that are T cells only, and see whether they have acquired or not the fluorescent telomeres from the APCs injected a day earlier. The answer is they do in large proportion. About 50% of these cells acquire telomeres in vivo in one night. We have also been validated by metaphysics spreads that these telomeres reside in the nuclei of the mouse T cells. And we haven't seen a single T cell of these CTV positive cells positive for the telomere signals that does not contain the APC telomere in the nuclei of the recipient T cells as yet, meaning that the incorporation is complete and they're not just stuck on the plasma lemma as some people thought. So again, this process is conserved in mice, but what is the importance of it? So to study the importance of the process, we have we've designed experiments in which naive OT2 T cells are allowed to form in vitro synapses and then sorted based on the fluorescent with APCs, obviously in the OT2 model with APCs that again have CY3 positive telomeres labeling, then derive two population of OT2 T cells so that they have either acquired or not the fluorescent telomeres from the APCs. They are transferred in two different sets of recipient animals. They are offered vaccination after 18 hours and then after 40 days again, a second boost and observed for an additional 50 days, making it a 90 days experiment. After 90 days and 2 rounds of vaccination, we discovered and demonstrated that the T cells that in the first in vitro reaction had acquired the telomeres from the APCs, has switched to a stem-like memory state in the spleen and in the lymph node. Also, there were increased numbers in those spleen lymph nodes. We characterized better the spleen in terms of phenotype. They switch towards stem-like and central memory state if they increase stem-like and central memory state if they had acquired the telomeres in the first round of synapse formation before the first cell division. So this is, as analogy, is essentially identical to what happens in humans, and this really proves what I said at the beginning of this presentation. So that the aging fate decision of T cell is really determined upon this single decision of telomere transfer. I'm not saying that there are not other mechanisms that can generate memory, but certainly this has been overlooked, and it's a very important one because the T cells that don't receive telomeres, even if they form memories, probably the quality of those memories, as we will show very soon, it's not the same quality of this phenomena I'm describing now. So the RAD51 that is present in the telomere vesicle is important for the longevity effect. What is the mechanism? Essentially, as you can see here, there is RAD51 in the vesicle. If I remove RAD51 in the vesicle and I transfer these vesicles to T cells, the colocalization is impaired. How did we do these experiments? We took, these are primary human T cells, we took resting primary human CD4s, we labeled telomeres of these T cells in green, and we got autologous APCs that we use throughout our CD3 depleted peripheral blood mononuclear cells, and silence RAD51 these APCs, label telomeres, get the telomeres out of the cell upon ionomycin stimulation, purify the vesicles and transfer them to these T cells with green telomeres, and after 24 hours study the colocalization between the green signal that is the telomeres of the T cells and the red signal that is the telomere of the donor cell. This is an important point because I read some skeptics that were saying that there are too many telomeres in these images. That's because only the green signals are the endogenous telomeres, and instead the red signals, as I said, are the ones offered in forms of vesicles. So the colocalization is impaired in the absence of RAD51, and so is the telomere elongation by Q phase, by metaphase Q fish. On the right. Moving slide again. RAD51 is important, as I showed before, in vitro for human T cell longevity or expansion, and in vivo for the longevity of the T cells as well. We have repeated the experiments essentially providing T cells with vesicles with or without telomere vesicles, with or without from APCs, with or without RAD51, and performed similar double over vaccination, and after 70 days and a resting phase of 30 days here, so making 70 days experiments, beautifully we show that the loss of RAD51 in the telomere vesicles completely abolished their longevity effect. So the transfer, so if I transfer vesicles without RAD51, the transfer doesn't happen. I'm not showing, but we know the reason as well. because people obviously were asking why the RAD51 of the T cells wouldn't compensate. The issue is structural. Essentially, RAD51 sits on the single-strand DNA, and when you remove RAD51 from these vesicles, we have demonstrated that the single-strand DNA in the vesicles is blunted. So essentially, you transfer vesicles that are similar in size, but they lack the overhang. So we think that the loss of the overhang is what basically makes it impossible for them to fuse a T cell chromosome ends upon transfer. And finally, we have investigated this, and we took this into a more clinical setting, in experiments in which we got T cells with or without telomere vesicles. Upon we first vaccinated animals with fluad. Fluad is a vaccine for influenza given to the elderly. We got fluad prime T cells. These T cells were then offered after 5 days, so at the peak of their effector phase in this animal model. They were offered vesicles with or without telomeres, and then transferred in another animal that wasn't immunized with fluid, and that animal was either immediately infected with influenza or the infection happened 15 days later. So the results were striking. So animals, so both cohorts of animals receiving are not telomere vesicles, but obviously fluid prime T cells were protected from immediate infections. Instead, animals as a control that receive here in yellow, animals that received T cells that weren't primed with the vaccine succumbed within 5 or 6 days. But the striking difference was that after 15 days, the situation was completely different. So only those prime T cells that received telomeres could support the animals upon delayed infection. So this is the demonstration that telomere vesicle transfer is clinically important, because it forms a functional memory T cells that persist and that can protect from later infections. That is the goal of every vaccination program. So we are really excited about this, because obviously we think that including vesicles containing telomeres or having ways to induce a telomere transfer reaction can form better long-lived memory T-cell responses or form new forms of prophylactic therapies of immune senescence and age. And moving slide again, at Centicell, we are taking now this into humans. We have proprietary compound which is called DOS. DOS are the disruptor of the SMAC. SMACs are the complexes, as I said, that regulate the senescence of the T cells. DOS are small cyclic peptides that bind to SMAC and destroy the SMAC. And they do a lot of things. I can't say much because this paper is under consideration now in Nature. But one thing I will share with you is that they induce the telomere transfer reaction. They do this in vitro, they do this in vivo, they do this not only in the immune system. So a clinical trial of the old will start next year that will initiate human rejuvenation by selective disruption of the SMAC through the DOS. And this is selective. Why is this selective? Because sestrins can be found at least in 2 separate macromolecular complexes. One was discovered by myself, and the other one was characterized by David Sabatini. So containing the other vectors, and sestrins were thought for a very long time to exert exclusively anti-aging function when acting through mTOR1 inhibition, but when sestrins are not in the mTOR and actually are in the SMACs, they have opposite pro-aging function, and we have now this compound DOS that has tropism only for senescent cells, and that binds exclusively to the SMAC, and that induces, it's the first drug in the world that induces the telomere transfer reaction in vitro and in vivo, and informs long-lived memory responses with or without vaccination. And that is what is really striking, that we can essentially offer the compound even without a vaccine, and animals are protective months later. A single injection is offered, which is amazing. So, in conclusion, we think that aging may be due to loss of telomere transfer reactions rather than simply telomerase inactivation, and we will have more exciting studies revealing different roles for telomerase very soon. I would like to thank all my team, Melania, Clara, Alessandro, Francesco, Federica, Andrea. We're all working together to solve this issue of aging in humans. And thank you again for the invitation. Thanks.
Wow, for your amazing research. And this is, I said before you arrived, that your work is really a game changer for so many things, for immunity and for rejuvenation. And it's amazing. So, my first question, probably the general public has, like, how good can you scale this drug? Like, let's say clinical trials go well. Um, and will it be a very expensive treatment? Will it be accessible for a lot of people? Or,
Well,
yep.
those are that's a difficult question for me to reply now because, as you know, there are matters of science and there are matters of business, right? So I probably cannot answer that question.
Yeah, okay. I understand. But
Yeah.
when people hear this, they want to know, like, when can I get get it and
Yeah.
how much do I have to pay? How much I have to save? Um but, so, um but yeah, um this is such an amazing outcome. And I wanted to ask you if you could use your principle also for other cell types in the body, or like, are you planning to maybe work on
We
that?
are
Because
doing it
I imagine you could
already.
use it for Parkinson and Alzheimer's and
Yeah,
so.
we are doing it already. Yes.
Oh, wonderful. So you're in the
Yeah.
process of in animals
Yes.
or in cells for now, or how?
Both. Both.
Oh wow, that's amazing. And
This is a phenomena that has been underlooked, and this is everywhere.
yeah, so so it will also be a drug. Would it be the same drug? Does it
Well, the
pass the
drug,
brain?
this drug, that's what we are figuring out now. It's something that is really important, obviously, because when you do these manipulations of senescence, something that we had really to make sure was the drug to be safe, not only effective, because there's always the risk when you do these manipulations of telomere signaling. So I'm pleased to say that in our large cohorts of animals, hundreds and hundreds of animals, we haven't seen cancer, we haven't seen autoimmunity, and we are very confident. Also, we can. This drug is very powerful, so we really need to use it in really, really small amounts, and apparently only once, to induce in animals long-term or what it seems permanent immune rejuvenation. So I'm not saying that there might not be treatments when we offer these drugs multiple times, but for sure there won't be short distance protocols because there are long-lasting rejuvenation cascades that are triggered by the treatment.
And another question, maybe Katie, I would like to also have this answered here. Do you think for people that have long COVID symptoms, do you think, I think this drug would be very helpful?
Yeah.
Are you thinking of that too?
Yes, yes, absolutely. Yes, for
And
sure.
with that, I pass the mic to Katie. Thank you.
Sorry?
Oh, I'll pass the mic
Oh,
to Katie if she wants to ask
yeah,
more about.
yeah,
Thank.
yeah.
Hi, it's Katie. Thank you so much, Dr. Lana. Really, really fascinating research that you're doing, and we greatly appreciate your time here. Also, anyone else that has any questions, please feel free to raise your hand and come up and ask questions, or have a look in the chat or type in the chat. There's a presentation pinned above and also links to the research. really fascinating stuff. And, building on what Katerina just asked as well, it's fascinating. And I know that you mentioned influenza and as Katerina brought up, COVID and long COVID. I'm wondering, is this something that could be used either potentially, and I understand, of course, we need clinical trials and why we should all support clinical trials, either as a prophylactic or as treatment for people that have had SARS-CoV-2. Thank you so much for your time.
Yeah, we the first clinical trial will be a trial of influenza vaccination of the old. But we also have data showing that the compound can be given without vaccination, and animals are protected from infections with influenza viruses. Months later, the single treatment is given. So definitely, this compound, for reasons that I cannot share, uh, offers a prophylactic arm to the immune system to face new challenges, and that cannot be restricted to viruses. This could be, for instance, transformative for cancer vaccines, in which essentially you could basically shake your T cell reservoir against things that the T cells didn't so before.
Yes.
Yeah,
Hi, sir.
sorry.
Yeah, I have a question. Thanks for your precious time. Just now you mentioned prophylaxis medicines like transmembrane
Sorry,
serotypes.
I can't hear anything. I can't hear anything. There is a
Yeah,
metallic sound. I can't hear anything.
your connection is really bad. If you could switch to a better spot, so we can hear you. Or would you like me to read
Hello.
one?
Can you hear me now? Hello.
Yeah, not.
That's much better.
Yeah,
That's
thank you.
better. Yeah.
So, yeah, as you just mentioned, the prophylaxis medicines like transmembrane serine protease, the enzyme inhibitors, if we use much, and those will affect lifespan of T cells or not, I would like to know.
Sorry, yeah, I can't hear anything.
Yeah,
Hello, am I audible?
I'm sorry, I can't hear anything. I
yeah.
can't.
In between, you break up. Do you want to ask? Maybe write in the chat, and then I read it out.
Okay, okay. And can you hear now? Hello.
Yes. Now, yes.
The enzyme inhibitors like transmembrane serine protease, TMPRSS2, if we use those enzyme inhibitors, those will affect the lifespan of T cells. That is my question.
I don't know the answer. If they block the transfer of telomeres, they will. Yes. But I haven't tested it, so I don't know.
Okay, sir. Thank you. Thank you.
Thank you.
Yeah, there's a question in the chat from Shiraz. If you, or if it's in general possible to also engineer telomeres in adults using tools like CRISPR. I think this is just a general question. Thank you.
Um, well, I guess with CRISPR technology, you can engineer really anything. Um, yes, in principle, that's doable.
Yeah, I don't know if you want to.
I wouldn't do it, and I'm not focusing on that. But in principle, I think it's doable.
Yeah, yeah, but so maybe do you want to point out? No, it's fine. The question, but why a drug approach is more beneficial, maybe for people that don't understand really, um, what the risks are of gene therapy and why you would rather prefer a drug approach.
Well, I guess that, I mean, we're talking about a drug that is given once, and I'm always skeptical with, permanent. It induces, by the way, it induces permanent modifications. This drug, it seems in the animals because it seems to induce permanent rejuvenation. So obviously that comes with permanent modifications. Um, um, but I think it's just much more manageable rather than playing with CRISPR.
Yes, I agree.
Yeah.
We don't have the risks enough, and it's precise, but also that precise. I think there's a lot of yeah. Um, the
There is a question on people. Well, we are starting the clinical trial next year, so we are manufacturing this drug now.
Yeah, that's really exciting. I'm really excited about that, and for purely egoistic reasons. Um, and so for reverse aging, are you planning on? Well, is it harder to start clinical trials and get funding for reverse aging, or maybe easier even? Um,
Well, I guess right now it's hard for everyone to raise fundings because it's such a bad time worldwide with the war and after the COVID. Also, the I think somehow also damaged expectations. When you go and talk to investors and people because people think the trials can happen in 1 year. Instead, that was an exception. So I, I think it has to be a bit kind of compensated somehow, and people need to be educated on what is actually standard process that in general a drug like this would undergo, right? But I'm confident that in the next 5 years, this could see the market.
is it an easier approach to maybe? Yeah, I don't know what your approach is for rejuvenation. Like people that are almost about to die, anyways. To get approval for that, maybe on a small
We
scale.
are focusing on people that are still relatively healthy because we are focusing on people who are 55 or 65 plus, probably 65 plus, because we're starting with Fluad, which is approved for 65 plus. This is the UK, so that makes sense. I think what we want to do is we want to add extra years and extra quality to life. That's what we are targeting. And in the field of vaccinations, so vaccinations obviously it's just the start. In the field of vaccination, there is really little available in terms of options for people to get immunized and to do that in a satisfactory fashion. And that relates to influenza, but that relates also to COVID 19 because people then have to get vaccinated many times. Don't really understand yet, probably at which cost. So, if you can reduce the number of vaccinations, so you can extend the longevity of and the quality of the response, and I think probably all the body benefits from that. So we look at we look at this in an holistic fashion, obviously.
Yeah, thank you for that answer. Joyce, did you have a question? You just joined it.
Yeah, hi. Very, very interesting. And thank you for your work. I was just, unfortunately, I missed some of it, but I'll just ask you about a couple of my favorite topics and you can tell me if it has any relevance. And that is like the diet and microbiome and inflammation. Thanks.
Yeah,
I'm done.
yeah. Well, talking about the diet, this thing is connected to metabolism a lot. So the beginning of the call, I mentioned that APCs need to understand whether the cells are worthy of a telomere donation or not. So antigen presence obviously is required, but that's not the only thing. And this was another point of controversy during the long reviewing of this paper, because people essentially were saying, you're showing that effector T cells have reduced capacity to get telomeres, but they are really powerful antigen-specific cells. So, and the answer I provided is that antigen presence is required, but it's not sufficient to trigger the transfer. And the full story is that we are hopefully showing it soon. T cells require fatty acid oxidation and antigen presence to get telomeres. So if they don't have adequate mitochondrial support, they cannot get the telomeres from the APCs. And we know all the story, we know all the pathway, but I can't show it now. But this paper is about to be submitted. And that's very important because you talk about diets, and obviously you know that during calorie restrictions you activate, and during fasting you activate fatty acid oxidation, and that has a benefit in terms of longevity. So we believe that this is the reason why intermittent fasting, for instance, can produce beneficial long-term longevity effects.
Oh, thank you.
No worries.
We are almost up to an hour, so I wanted to check if you have time for one more question.
Yeah.
If yeah, okay. Nardish, you joined the stage. Do you want to ask a question?
it's it's more of just a like a question for me. I didn't miss the whole thing, so I'll go back and listen to the replay. But I'm curious about how telomeres are studied. Like, what technique do you go through to study telomeres?
Yeah, we have used an array of techniques. We have used standard fish. We have introduced telomeres in live cells, as I mentioned at the beginning, with glass beads, because this allowed us to preserve cells, donor APCs, in a viable state. That is essential then to study the movements of things when you do cell-to-cell cultures. We have used flow fish, so studying basically telomeres by flow. We have used telomere restriction fragment analysis. We've used universal stelas. We have used qPCRs. We have used dot plots. Yes, pretty much everything we've used.
Okay, so well, if I can have time for another follow up,
Yeah,
you mentioned fish, and while I'm I'm no expert, I'm an investor. So my question comes back to I'm from my understanding, and I hope you're familiar. Bionanogenomics has this new device that's out that
sorry. I can't understand. Sorry.
okay,
Yeah.
um, okay,
Um,
I'll just end.
okay. Should I just read what's in the chat? If you know about bionanogenomics and their optical genome mapping, Saphir machine for a sequencing
Oh,
technique.
sounds exotic. I don't know it. I need to read about it.
Okay, yeah, great. I think it's a sequencing machine that
It's a
yeah.
It well, kinda. I don't want to interrupt. I'm sorry, but it's not. So they don't classify themselves as a sequencing company. They classify themselves as a new category because of their technique. I don't want to get into the specifics of it, but if I can just try to simplify it in my non uh, professor type voice. Well, it takes a DNA and um, strips it down to strands that it can then process into nanochannels and use some sort of light to determine what the signature is, which is different from like taking a gene and then smashing it into a whole bunch of pieces, which is kind of the standard technique for sequencing today. We sort of take those smashed pieces and put the puzzle back together, but BioNano sort of just kind of reads it in a linear fashion instead of that puzzle piece deal. And that's the best that I can do. But if you're not familiar, please check out BioNano and follow me because I'd love to talk to you afterwards.
Absolutely, absolutely. Yes, we can stay in touch. You probably do. You have my details because I don't have yours, so
Ah,
it would be
okay.
difficult for me to.
I'll follow the I don't know, maybe a back channel. I'm driving at the moment. I would send you my
Yeah.
email, but please hang around. I'll be stopped in a moment, and we can discuss.
Yeah.
So the I'm reading the website. It's a high speed, high throughput optical genome mapping and structural variation detection for human and clinical research. So they apparently they look at the sequence, but also at structural changes, but in an optical way. But it's really not anything really new, anyways. But yeah, we use optics for a lot of stuff nowadays. But yeah. Thank you so much for taking the time out of
Oh,
your busy
nice. Thank you.
schedule.
Very
It
exciting.
was
Thank you very much.
it
Very
was an
glad. Very
honor having you here, and your research is so exciting. Maybe next year when you start clinical trials, you can come back one day. That would be really
sure.
great.
Sure. There'll be more exciting things you'll see soon.
Yes, I'm really glad. Just probably I say it in the name of everyone that you do this research and that you're using your brain to do this, because this will be very helpful for all of us. We are all going to be sick and aging.
That is true. That's true. I've turned 26 this week, so that's
Yeah,
we're
I
all
have a cold
interested.
right now. I have a cold right now. If I could skip this once a
Me
year.
too. Actually, I apologize for coughing, but I got a cold a couple of days
I
ago.
have the same thing. I'm coughing too. It's so annoying. And yeah,
Thank
so
you. Thank you
I'm
so much.
waiting for your drug.
Thank you. Very kind. Thank you.
Thank
I
you.
sent you a message in your back channel. It's my email. If you could verify that you received it, because I don't want to lose contact with you.
I didn't receive the message.
Yeah, you don't follow each other, so it's probably in a request. So if you go on that chat, well, it's on your app all the way on the top. There's a little paper airplane signal, and then if you go there, they should be popping something up back channel, and then there's chats, and then there's a button on top requests. So there's where things land if you don't follow each other, and if you click on
You can
that.
just, if you have LinkedIn, you can just write me. I'm Alessio
Yeah,
Lanna on LinkedIn, so it's very
yeah.
easy.
So
It's
the
the first
info.
time for me on Clubhouse, so I didn't even know about this app. So it's quite so in the US must be very popular, right? So I never heard of this.
Yeah, especially during COVID, it got really popular, and now it's kind of like
That's
everything.
great. Never heard before. Okay, cool.
Yeah, so Nerdish, I shared in the chat the website of Alessio's company, and there's also his LinkedIn information and so on. So just reach out there. Okay, perfect. And thanks
Thank
everyone
you.
for
Thank you
coming.
so much. Thank you.
Thank you.
Thank you,
Thank you.
Katerina and Alicia, very much. It's great.
Okay, I'll close the room. Oh, right. Follow the club if you like discussions like this. We'll have tomorrow at 9 p.m. EST Dr. Dumas talking about multi-level development of cognition in AI. And then on Friday, we'll have Dr. Frank talking about intelligence as a planetary scale process. I think that was really interesting, his work. So feel free to come back. I hope to hear you all soon. And Alessio, all the grants, all the funding for your work, please. I wish you well.
I'll
Okay.
get you talking to my investors. Thank you. Bye.
Okay. I
Thank
don't
you.
know if your grandmother does this, but in Portugal they put a little candle on some saints. Ask your grandmother to do
Yeah.
that. Okay, bye.
Thank you. Ciao. Thank you. Thank you.
Have a
Bye.
great day. Bye, everyone.